Very good evening, students. Today we are going to start with the previous year question papers from today, okay. So yes, today we are going to discuss 2019 NEET Botany paper. Clear? And I have picked out all the questions which are applicable to our NEET UG syllabus 2025. Only those questions we are going to solve now. Okay, yes beta. Yes, if you look into the first question, it is from the anatomy of flowering plants. The very first question is from anatomy of flowering plants, which is from the monocot leaf, okay, and this is from bulliform cells. Grass leaves curl inwards during very dry weather. Select the most appropriate reason from the following, he is saying, okay. So yes, if you see monocot leaf, okay, in a monocot leaf, over the epidermis you will have large cells. These are called as bulliform cells. These are what ma? Bulliform cells. Whenever water conditions are more, whenever there are high water conditions around, these bulliform cells will absorb water. It will absorb water and keep the leaf turgid. It will keep the leaf turgid. During dry conditions, whenever water is not available, in those conditions the bulliform cells will curl the leaf inwards and it will make the leaf flaccid, okay. In the absence of water or in dry weather conditions, the flaccidity is shown by bulliform cells by curling the leaf inwards. So here if you look into the option beta, for grass leaves are what? Monocot leaves, right? Monocot leaves. So grass leaves will respond to dry conditions by curling inwards, that is the flaccidity of bulliform cells. Flaccidity is shown by bulliform cells. Clear? Second question if you look into, this is from ecology, where he is saying, asking you on ex situ or in situ conservation, very important concept, wherein which of the following is not a method of in situ conservation. If you see biosphere reserve, that is an in situ conservation, wildlife sanctuary is an in situ conservation, sacred grove is an in situ conservation, whereas botanical gardens which are man-made, these are ex situ conservation. Botanical gardens or zoological parks, they come under what beta? Ex situ conservation. Very important session ma, where we are going to exercise the concepts and solve the PYQs. Don't miss at all, okay. So third question if you look into, this is from cell cycle and cell division. This is from cell cycle and cell division, wherein different shapes of the chromosomes. This is from different shapes of the chromosome. The question is from metacentric chromosome, okay, wherein submetacentric chromosomes, the shorter and longer arms of submetacentric chromosomes they are asking, okay, shorter and longer arms of submetacentric chromosome. What is a submetacentric chromosome? The centromere will be close to the middle, which is forming one short arm and one long arm. Short arm is said as P arm and long arm is said as Q arm, okay. Now we will exercise the different shapes of chromosomes as well. If you consider different shapes of chromosomes, different shapes of chromosomes, okay, different shapes of the chromosomes, you can see clearly during which phase, Nana? Anaphase. During anaphase of the cell division, you can clearly see different shapes of the chromosome. If the chromosome is having two equal arms if it is having, we call such type of chromosome as metacentric chromosome. What do you call it as beta? Metacentric chromosome. This shows out to be a V-shaped chromosome. This shows out to be a V-shaped chromosome. And if you have one short arm and one long arm, this is called as submetacentric chromosome. What is it called as ma? Submetacentric chromosome. This gives out to be an L-shaped chromosome, okay. Then comes acrocentric chromosome, acrocentric, which gives out very long arm and very short arm. This will be of J shape. This will be of J-shaped chromosome, okay. And last but not the least, that is telocentric chromosome. Telocentric chromosome will give one very short arm, one very small negligible short arm and one very long arm. This gives out to be an I-shaped chromosome. As such, during anaphase of cell division, you can visualize 4 different shapes. One is V shape, that is metacentric, L shape submetacentric, J shape acrocentric, I shape telocentric. The short and long arms are clearly visible in submetacentric and acrocentric chromosomes, where shorter arm is called as P arm, longer arm is called as Q arm. So, the correct option is 2. Okay? Shorter arm is P and longer arm is Q. Now, the next concept is about respiratory quotient. This is from the chapter respiration in higher plants. Okay? Respiration in plants. So, here if you keenly look into beta, what is RQ value? What is RQ value? RQ value is the amount of carbon dioxide evolved by the amount of oxygen consumed. Okay, for different respiratory substrates. Okay, for different respiratory substrates. Once let us recall all the RQ values of different respiratory substrates. Okay, respiratory substrate and RQ value. If you take carbohydrates, that is glucose, we have 6 carbon dioxide and 6 oxygen molecules are utilized, wherein RQ value will be 1. For fats and proteins, do remember beta, the RQ value is less than 1. Because more amount of oxygen is utilized for the CO2 to be evolved. So, that here for fats it will be 0.7, for proteins it will be 0.9. Okay. When it comes to the respiratory substrate of organic acids, for organic acids it is more than 1. For example, oxalic acid, it is around 4. Okay, oxalic acid it is around 4. As such, malic acid will be around 1.3. Then what will be the RQ value? For anaerobic respiration, that is for alcoholic fermentation. In alcoholic fermentation, we all know that 2CO2 is evolved along with ethanol, 2 CO2 are evolved and there is 0 oxygen that is consumed. That is why it is anaerobic respiration. Something by 0 is always infinity. Something by 0 is what, Nana? Infinity. So, these are different RQ values where you have to remember. This is highly important. Okay. So, yes, here the RQ value is asked for tripalmitin. Tripalmitin, that is a fat. Okay. Tripalmitin, which is fat, that is option 2. Okay. 0.7. Then this question is from microbes in human welfare, wherein which of the following is commercial blood cholesterol lowering agent? That is statin. Okay, statin is the commercial blood cholesterol lowering agent. Again, one more question from cell cycle and cell division. Okay, wherein the different phases of cell cycle are asked in a sequence. If you clearly see in a cell cycle, just a second. Yeah, in a cell cycle we have 2 phases, that is interphase and M phase. Interphase has 3 subphases, that is G1, S and G2 phases, and M phase followed by it. Okay, M phase is called as the dividing phase, division phase. Okay, M phase has again karyokinesis and cytokinesis in it, where karyokinesis has prophase, metaphase, anaphase and telophase. Clear? So, what will be the sequence? G1 followed by S followed by G2 followed by M. G1, S, G2 and M. Option 4 is the correct answer. Next question, if you look into, the question is from Cell the Unit of Life, where the organelle based question is asked. If you clearly see, which of the following statements is not correct, he is saying with respect to lysosomes. We all know lysosomes have numerous hydrolytic enzymes, correct, like lipases, proteases, carbohydrases. These are all what ma? Hydrolytic enzymes, correct. Statement 1 is true. Next, the hydrolytic enzymes of lysosomes are active under acidic pH, true. They are inactive under basic pH, they are active under acidic pH. Lysosomes are membrane bound structure, correct. These are single membrane bound structures which are in endomembrane system, clear. And if you look into fourth statement, lysosomes are formed by the process of packaging in endoplasmic reticulum, wrong. Lysosomes are formed because of the packaging in Golgi bodies. Packaging in what ma? Golgi bodies, that too from the trans phase of Golgi. From trans phase or concave phase of Golgi, lysosomes are released in the form of vesicles. Lysosomes are released in the form of vesicles. Okay, it is from Golgi bodies, not from ER. So, statement 4 is wrong statement. Whenever statement based question is given to you, you have to look into whether he is asking you to choose on the correct statement or incorrect statement. Just don't go in a rush of choosing the option without reading the question properly. That is the kind reminder I give you to all. Next, here if you see, this is from sexual reproduction of flowering plants, wherein which of the following statements regarding post fertilization development in flowering plants, again it is incorrect. Incorrect statement he is asking you. Ovary post fertilization will turn into fruit. This is a post fertilization event. Zygote will turn to an embryo. This is also a post fertilization event. Central cell or polar nuclei will turn to triploid endosperm. This is also a post fertilization event. Ovules turning to embryo sac is a pre-fertilization event. Okay, ovule develops into embryo sac is what ma? Pre-fertilization event. So here what will be our answer? Option 4 is the correct answer. And next, this is from biomolecules, wherein concanvaline A is what? Okay, this is a lecithin. That is why when I am dealing with biomolecules also I have said you, those charts are very much important from biomolecules. Each and every chart, look into it more keenly. Okay, whatever tables are given in the biomolecules, definitely questions will be arised from that. You have to look into it more keenly. Okay. Now, again statement based question from biomolecules. If you clearly see, first statement, coenzymes and metal ions, they are tightly bound to the enzyme protein and is called as prosthetic group. Wrong. Beat coenzyme or metal ion, coenzyme or metal ion or prosthetic group all the three together, they are called as what? Cofactor of the enzyme. They are called as cofactor, not prosthetic group. This is wrong. Next statement, if you see, a complete catalytic active enzyme with bound prosthetic group is called as apoenzyme. Wrong. Apoenzyme means what ma? The protein part of the enzyme is called as apoenzyme. The protein part of the enzyme is called as apoenzyme. Non-protein part can be cofactor. Cofactor can be anything. Coenzyme, metal ion, prosthetic group. Clear? So yes, this statement is also wrong. Both statement 1 and statement 2 are false. Okay. So option 3 is our answer. Next question, if you see, this is from molecular basis of inheritance or MolBio. Purines found between DNA and RNA. DNA and RNA have same set of purines. Those are what ma? Adenine and guanine. Adenine and guanine, they have same set of purines which are heterocyclic. But when it comes to pyrimidines, pyrimidines will be different beta. Okay. For RNA it will be uracil in the place of thymine. In DNA we have thymine, whereas in RNA we have uracil. Okay. Uracil will uracil or thymine will bind to what? Adenine, okay, and we have guanine, sorry, cytosine, which binds to guanine here. Here also it is cytosine. So what are the two pyrimidines that are present in RNA? Uracil and cytosine. Here in DNA we have thymine and cytosine. That you have to keep it in your mind, okay. Next, this question is again from Cell the Unit of Life. That is why I always say you Cell the Unit of Life or cell cycle, cell division more important, and plant physiology, which covers more number of questions actually. Which of the following pair of organelles does not contain any DNA? Both the pairs of organelles should not contain the DNA. That is his question. Here if you see, mitochondria has its own DNA. That is why that is called as semi-autonomous or self-replicating. Mitochondria and chloroplast, both of them have DNA. Nucleus also has DNA. So these three options we can eliminate because one of that organelle has DNA. Clear? But then lysosomes and vacuole, they don't have their own DNA. These organelles, they don't have DNA. Here option 3 is the correct answer. Next, if you see, this is from again genetics, wherein he is asking you the question from sex determination, okay. This question is from sex determination. Wherein male fruit fly is heterogametic, yes, because it has XY chromosomes, okay, and in male grasshoppers 50% of sperms have no sex chromosome, yes, only 50% of them will have X chromosome, 50% will not have X chromosome. In domesticated fowls, sex of the progeny depends on the type of the sperm rather than egg. Wrong. In birds we can see female heterogamety. In birds we can see what ma? Female heterogamety. Okay, it is not male heterogamety, it is female heterogamety in bird. So yes, it is egg that is deciding the sex of the individual in the birds, not sperm. Okay, they have ZW. Okay, heterogamy you can see in the females, not in males in the bird. So here the incorrect statement again is option 3. Okay, next. Match the following type of question from microbes in human welfare, wherein lactobacillus is used in the preparation of curd. Saccharomyces cerevisiae, that is yeast, is highly used in the preparation of bread. Aspergillus niger is used to give the citric acid. Acetobacter acetifer, acetic acid. So here if you clearly look into the options, A is 2, B is 4, A is 2, B is 4, C is 3, D is 5. Option 2 is the correct answer. Next question, if you see ma, what is the direction movement of sugars in phloem? In xylem, okay, if you see, in xylem, the movement of water is from roots or root hairs to the tip of plant, to the tip of shoot. Here you can see unidirectional movement. This is a unidirectional movement. If you see phloem will transport sugars from source to sink. Okay, source to sink. This is a bidirectional movement. Bidirectional movement. Source can be mature leaf. Source can be a mature leaf. Sink can be anything like a root or a young shoot bud. Shoot bud. Okay. So it is from source to sink. It takes place in a bidirectional way. So here the answer is 4. Of course, transport is not there for our syllabus, that is fine. Now if you see, again this is from sexual reproduction of flowering plants. In some plants, the female gamete develops into embryo without fertilization. Without fertilization, female gamete developing to the embryo, you call it as parthenocarpy. If it is in animals, it is parthenogenesis, but if it is in plants, it is parthenocarpy. Persistent nucleus in seed, persistent nucellus, even after seed formation, if you can see nucellus, it is called as perisperm. Persistent nucellus is what ma? Perisperm. Best examples are beet, black pepper, castor. These are the best examples. Next, it is from it is from gene mapping, wherein what map unit is adopted in construction of genetic maps. Okay, if you see, it is given by Alfred Strudworth, wherein genetic map, what will be the flow? If we see a unit distance between two expressed genes is representing 10% crossover? No. A unit distance between two expressed genes represents 100% crossover? No. A unit distance between two genes represents 1% crossover. Unit distance is represented as centimorgon, 1 centimorgon. Okay, between two genes, in between two genes, it is 1 centimorgon. It is 1 centimorgon or 1 map unit. Okay, so here statement 3 is the correct statement. Okay, and next if you see, which of the following statements regarding mitochondria is incorrect? Again, incorrect statement we have to look into. Okay, incorrect statement we have to look into. Outer membrane is permeable to monomers, carbohydrates, fats and proteins. True. Enzymes of electron transport are embedded in outer membrane. Wrong. All the enzymes, electrocarriers, channels, complexes, whichever are required for electron transport are embedded in inner mitochondrial membrane. They are embedded in what ma? Inner mitochondrial membrane, not on the outer membrane. And outer membrane is totally permeable, whereas inner mitochondrial membrane is not completely permeable. Okay, it has many enfoldings called as cristae. Mitochondrial matrix consists of single circular DNA and ribosomes. All of it is correct. Once let us recall the mitochondria structure, wherein outer mitochondrial is completely permeable, inner mitochondrial membrane has enfoldings called as cristae, and all the electron carriers, protein complexes and transporting molecules whichever are necessary for electron transport are present in inner mitochondrial membrane. And this space whichever is there, this is called as intermembrane space. What is it ma? Intermembrane space, and inside it is mitochondrial matrix. Clear? This is the structure of mitochondria. And then if you see cell in G0 phase, it will exit the cycle. Okay, let us recall its ones. If this is the cell cycle, G1, S, G2, G1, S, G2 are the phases of interphase, and then we have M phase. If any cells like nerve cells or heart cells, these cells will rarely conduct the cell division. So, they will exit the cell cycle from G0 phase, G1 phase to G0 phase. Okay, few cells like brain cells and nerve cells, they will skip the cell cycle from G1 phase to G0 phase. From G1 phase to G0 phase. Again, whenever they are resuming back to the cell division or cell cycle, they will come from G0 phase back to G1 phase only. Okay, keep that in your mind. Fine ma. Next question. It is from morphology, where from placentation and aestivation, that and all you should not leave at all, you should not skip at all. It is very important. Either from placentation or from aestivation, definitely questions will come. Placentation, in which ovules develop on the inner wall of the ovary. From inner wall of the ovary, it is parietal placentation. From inner wall of the ovary, ovules developing from the inner wall of the ovary, that is what ma? Parietal placentation, which leads to a false septa or replum formation. False septa, which is also called as replum. Okay, this is seen in parietal placentation. Best example for parietal placentation is the family Brassicaceae. Okay, mustard, brassica, this and all are the examples of parietal placentation. Clear ma? Next, conversion of glucose to glucose 6-phosphate. The very first step of glycolysis, which is the irreversible step. Okay, first irreversible step of glycolysis is catalyzed by what? Hexokinase. It is catalyzed by hexokinase. And then, if you see, what is the fate of male gametes once it is discharged to synergids? Of course, one male gamete will fuse with XL, and the other male gamete will fuse with central cell. The other male gamete will fuse with what ma? Central cell. Okay, one male gamete, haploid male gamete fusing with haploid egg, it will form a diploid zygote. It will form what ma? Diploid zygote. Okay, and next, haploid male gamete fusing with diploid central cell, it will form a triploid endosperm. Okay, this is called as syngamy, and this is what ma? Triple fusion. This is triple fusion. Syngamy and triple fusion together, we'll call it as double fertilization. So here what will be our answer? One fuses with the egg and the other fuses with the central cell. Option 4 is the correct answer. Next question, what is the site for perception of photoperiod necessary for the induction of flowering? Always perception of light during photoperiod, it is by leaves. Okay, leaves will perceive the light. Then select the correct, this is from Living World, where the nomenclature of Carolus Linnaeus binomial nomenclature, from that characters or the components or the rules of binomial nomenclature, from that the question is asked, wherein scientific name should have one generic name and one specific epithet. Gene should have one capital, genetic name should start with capital letter, and specific epithet should be with the small letter. Okay, specific epithet should be in small letter. As they are printed, they should be in italics, and scientist name after that should be in bold or Roman. Okay, so here the correct option will be 2. Most of them will be confused looking all the 4 similar only, but not like that beta. You have to check on whether generic name is starting with capital letter, species name is starting with small letter, both of them are in italics or not, and if scientist name is given, if it is italics, then it is wrong. It should be bold or in Roman. Okay, that you have to make a note, and then you can identify the correct word. Okay, yeah, this is from Plant Kingdom, wherein pinus seed cannot germinate and establish without fungal association. This is because of obligate association with the roots. Okay, pinus roots are associated, pinus has taproot system, they are associated with fungi. Okay, they are associated with fungi to increase its network and to absorb more water and nutrients. So this is an obligate association, so there is no skip of it, because that is an obligate association with them. Next, which of the following features of genetic code does allow bacteria to produce human insulin in recombination DNA? This is from biotechnology, wherein from bacteria or it human being, the genetic code is universal, wherein AUG codes for methionine only, UUU codes for phenylalanine only, be it is in bacterial cell or it is in a human cell. As genetic code is nearly universal, because of this feature, we can conduct the genetic rDNA technology will work on this, even if it is in bacteria or in human. Okay, this is how a bacteria is injected to produce human insulin by rDNA technology. It is because of only one feature where genetic code is nearly universal. Next, which of the following statements is correct, he is asking. Viroids, we know lack of protein coat is viroid, this is correct, and prions consist of abnormally folded proteins. Always do remember beta, prions abnormally folded proteins, it is rhyming, right? Prion protein, like that you remember. Only abnormally folded protein without any genetic material is prion. Viroids has only genetic material, that is RNA. Okay, best example is what disease? Potato spindle tuber disease. Okay, so here viroids lack protein coat. Obligates, viruses are obligate parasites, this is also true. Okay, virus is a obligate parasite. Infective constituent in virus is protein coat, wrong. In a virus, infective constituent is genetic material. It is what, na? Genetic material, be it single stranded or double stranded DNA or RNA. Okay, in virus the infectious material is genetic material. And next, if you see, which of the following statement is incorrect? This is from fungi. Okay, morels and truffles, which belongs to what class? Ascomycetes. Ascomycetes, they are edible delicacies, true. Claviceps is a source of alkaloids, this is also of Ascomycetes, true. Conidia are exogenously produced asexual spores, and ascospores are sexually produced, they are sexual spores which are endogenously produced. Yeast also belongs to Ascomycetes, but this is unicellular. Yeast is unicellular, it is not multicellular, it does not have any hyphae or filamentous bodies. So, as he is asking you incorrect statement, the option is 4. Okay, and then match the column 1 and column 2. Saprophyte means what beta? Decomposition of dead organic material. Okay, saprophytic nutrition is decomposition of dead organic material. Parasitic nutrition means living on living plants or animals, that is parasitic nutrition. Lichens is a symbiotic association between algae and fungi. Mycorrhiza is a symbiotic association of fungi with roots of higher plants like pinus. So, here if you clearly look into option, A is 3, A is 2, B is 3, C is 4 and D is 1. Option 4 is the correct answer. And then this is also from genetics, wherein the frequency of recombination between gene pairs on the same chromosome is a measure of distance between two genes is explained by genetic mapping or chromosomal mapping is developed by Alfred Sturtevant. Okay, wherein distance between two genes on the same chromosome, okay, distance between two genes on the same chromosome, he said it as 1 centimorgon, as how he has developed a genetic map. What did T.H. Morgan do? He has conducted an experimental proof for chromosomal theory of inheritance. Sutton and Bovary, they have proved the chromosomal theory of inheritance, and the experimental proof is given by T.H. Morgan. Gregor John Mendel, he is the father of genetics. This and all you have to be very clear with respect to the scientist from genetics. So, here the option is 3. Now, the next question is match the following type of question from gene expression. This is from what, Nana? Gene expression, wherein I gene is a repressor, Z gene is what? Beta galactosidase, it codes for beta galactosidase, A is for transacetylase, Y is for permease. Okay, if you take the mRNA strand from promoter region to terminator region, After promoter there is operator, and then it is followed by 3 structural genes. It is followed by what? 3 structural genes, that is Z, Y, A in the Lacoperon model. Okay, from 5 prime to 3 prime, wherein I gene which is present in the operator region, it will release repressor. It will release repressor. Okay, of these 3 structural genes, Z will code for beta galactosidase, Y codes for permease, and A codes for transacetylase. So here A is option 3, B is 1, C is 4, and D is 2. Option 3 is the correct answer. Next question, if we see beta, this is from phytohormones or plant hormones. It takes very long time for pineapple plants to produce flowers. Application based question, look carefully. Which combination of hormones can be applied to artificially induce flowering in the pineapple? Of course, to induce flowering, especially female flowering, it is with respect to 2 hormones, that is auxin and ethylene. Okay, it is auxin and ethylene. Clear ma? So here the option is 1. Next, the concept of omnis cellula, e cellulae, this is from the modified cell theory. This is given by Rudolf Virchow. And then this is from plant kingdom again. Phloem in gymnosperms. In gymnosperms, we don't have, from xylem, it doesn't have vessels. Vessels are absent. From phloem, it doesn't have sieve tubes and companion cells. Instead of sieve tubes, it consists of sieve cells. Instead of companion cells, it has albuminous cells. Albuminous cells. Clear? So yes, here phloem in gymnosperms will lack both sieve tubes and companion cells. Instead of sieve tubes, it has sieve cells. Instead of companion cells, it has albuminous cells. The next is the same question. We have discussed it just now. Now, the last question. It is from the genetics again. See, from genetics of 12th NCERT, we can find again huge amount of questions, wherein here one strand is given and he is asking under which of the following conditions there will not be any change in the reading frame of the mRNA. The reading frame of the mRNA will not be changed when the 3 sets, okay, 3 nitrogen bases are shifted, not one at any position, or by insertion or deletion of one, the entire reading frame will be changed. By insertion of 2 nitrogen bases, it will be changed. By either insertion or deletion of 3 nitrogen bases which codes for 1 codon, right? So yes, by insertion or deletion of 3 bases, the reading frame will remain unchanged. Here it is at 7th, 8th, 9th position, GGU. If that is deleted, the reading frame will not be changed. If it is either insertion also, there will not be any change in the reading frame. Clear? So this is all about 2019 NEET paper beta. In coming class, we will look into 2020 NEET paper as well. Like that, we will do up to 2024. These sessions are highly useful for all NEET aspirants. So stay tuned. Thank you.